Good afternoon, everybody. I'm Andrew Creek. I'm a Citrus Development Officer with New South Wales Department of Primary Industries and Regional Development now. Look, I'm co-hosting this event today with Emily Pattison. You may see her there on the screen. And this webinar is an output today from that Citrus Industry IPDM Extension Program. It's been a collaborative project for the last 4 years with New South Wales Government, Queensland Government, Western Australian Government, also Biological Services, Riverina IPM. And the project was funded by Hort Innovation and your Citrus R&D levy. At the start of that project, we did a bit of a survey, and Phytophthora was mentioned as one of the more important diseases. And I'm not sure if it's for Emily, but also certainly for me when I'm called out to growers' farms, have a look at my sick trees, what's wrong here, get the shovel and have a dig and say, yeah, we've got some problems here with these roots. So it's good that we are having something in our extension program for roots and soil-borne diseases. And today we're joined by Dr. Tony Pattison and Dr. Nerida Donovan, who will introduce a little later, that will give some presentations. I'll hand over to Emily Pattison, our co-host now.
Thanks, Andrew. So as Andrew mentioned, I'm Emily and I'm co-hosting this webinar. So I also work as a Citrus Extension Officer, similar role to Andrew, but I'm based in Nerida in far north Queensland. So I'm going to introduce our first speaker, Dr. Tony Pattison. So Dr. Pattison has migrated into soil health from a background in nematology. He is based in Innisfail, which is well known for its rainfall, which is about 3.5 meters a year for anyone who's wondering, and its banana production. So Dr. Pattison has worked in many crops dealing with soil health and nematology issues, but banana soil health has dominated his career. He's spending a lot of his time working in soil suppression of the infamous Panama disease and looking at ways to use agroecology and natural soil suppression to combat the disease. So while bananas are a fairly unique horticultural crop, Dr. Pattison has found a lot of the principles he is researching are transferable to disease suppression and soil health in other crops, which is why we have asked him to talk here today. So thank you very much, Dr. Pattison, for joining us. Yeah, take it away.
Thank you very much for inviting me to come along today and to talk about soil health. And as Emily said, I've been working on soil health for the past 30 years up here in North Queensland. And I'll say from the onset that my knowledge of citrus production systems is very limited, and this presentation will draw a lot on the work that I've been involved with bananas. But I believe many of the concepts are the same when it comes to soil management and its impacts on some of the principles of how to overcome those losses of soil health. So just before starting, I'd like to acknowledge all the First Nations people and pay my respects to elders past, present and emerging. And I'm on the land of the Mammo people in North Queensland. So we're just going to take a step back into the past to start off with. And soil health used to be considered an easy fix. Just add more fertilizer. And if you had a problem, just add more fertilizer. And this is what was depicted on this billboard from many years ago. The hint to successfully to grow your crop is just add soils as a growing medium for fertilizer. And I hope this is no longer your thinking about soils. And through this presentation, I really hope to show you why. And we're going to start this off with Emily's going to launch a poll about some of your thoughts about soils. So Emily? So the question is, which of Earth's biospheres has the greatest biological diversity and biomass? Is it the oceans, the tropical rainforests, soils, freshwater systems, deserts? So if you want to add what you think is what's the most diverse systems, we'll just give a few minutes there. So most people have said soils. There's a few people that have said oceans and tropical rainforests, but yes, the correct answer is soils, because that's what we're talking about. And actually 40% of all organisms spend part of their life in the soils, which contains about 1/4 of the biodiversity found on Earth. organisms under the surface easily outweigh all the animals found on the surface. Soil organisms comprise about 17% of all biomass found on Earth, and that's around 93 billion tonnes. Plants, which is the other half of the soil, represent the greatest proportion with around 80% of living creatures. So between plants and soil animals, you've got 97% of all life on Earth. So why talk about soil health? Well, as I said, soil is a living entity and it sustains plants, animals, and people. The advances in soil science and soil biology have shown us just how important soils are and how the different organisms interact with one another and have very different properties. Many of the soils used in agriculture are degrading and we are losing more good agricultural each year. But what we are still missing in some of the cases is how we can draw those links between agricultural practices, those soil biological interactions, and the healthy soils that lead to sustained crop productivity. So what is soil? Let's start off by answering that question. Around 50% of the soil is made up of inert materials, that is, the minerals derived from weathered rocks, and it gives the soils their physical properties. Next, there is air and water, and these are interchangeable. So as the soil dries out, there becomes more air and less water, and vice versa. As it gets wetter, there's more water, less air. There is only a small proportion, only around 5%, is organic matter. And this really has a big impact on soils, influences the physical properties, like how compacted a soil is, the chemical properties, like how much nutrients the soil can hold, as well as the biological properties of which organisms are present and what they do. But there are also a lot of other terms used for soils, so I'd like to explain a bit about these before we move on. Firstly, at the bottom of the screen, I'm just going to get my highlighter here, is soil nutrition. And this is pretty self-explanatory. It's about the nutrients in your soil and what's important for your crop production. Next is soil fertility. This is considered all the nutrients plus the other soil chemical properties like soil pH and the nutrient holding capacity. Next, if we move out again, we have soil quality. And this considers the fertility properties of the soil, but as well, it takes into account some of those physical properties, like how compacted that soil is. Then as we move out again, we come into soil health. And this considers all the previous things, but looks at more detail, the biological properties and how they interact. And soil health considers how problematic your soils are in producing crops, but also determines the condition of the soil and the condition of the soil on your farm that you hand on to the next generation. And that is what we'll be discussing today. After that, at a big picture level, we have soil security, which is more about soil conservation and preventing degradation at a larger scale. So let's start off our soil health journey by taking a handful of soil. And what you may see are only soil particles, but it is something that is full of life. Let's start off with the smallest organisms, the viruses. The soil is full of viruses. There are billions of virus particles, and the science of understanding these is only really relatively new. But they're not technically living, I guess, virus particles, but they have a very important role in regulating soil biology. Next, in that handful of soil, you may have a couple of 100 billion bacteria with as many as 1 million different types. And a fun fact, the weight of soil bacteria in Earth's soil makes up about 12% of all biomass of living organisms on the planet. More than 10% of living on this planet of Earth is a bacteria. As we move on, you will also get a diverse range of fungi and perhaps up to 100 meters of hyphae in that handful of soil. And another fun fact is that the fungus Armillaria ostoia is the largest living organism on Earth. The fungus, known as the honey fungus, is found in the forests of Oregon in the USA, and it is estimated to cover around 965 hectares. That is about 1,300 football fields or nearly 10 square kilometres. And it's estimated to be nearly 2,000 years old, over 2,000 years old, I should say, and could possibly be up to 8,000 years old. It appears above the ground as many mushrooms, but below the ground, it's one clonal network of a genetically identical mycelium. As well as microorganisms, you will also get microfauna, microscopic animals, things like protists, which include amoeba, and you get several thousands of these. The other very important microfauna are soil nematodes, and personally, these are my favourites as I have a nematology background. Again, there are several thousands of nematodes in a handful of soil. Nematodes are a very important indicator of soil health, as there are good nematodes and there are bad nematodes, and we'll talk a lot more about them during this presentation. As the organisms get bigger, you may see some mesofauna like mites, and there may be several hundreds of these, or other mesofauna like collembola, which are better known as springtails. And if you're lucky, you may even find some macrofauna, the big animals, the earthworms, and these are much easier to see, as are the other types of macrofauna that inhabit the soil, things like ants, spiders, millipedes, and isopods, such as this woodlouse. And as I said before, around 40% of all life on Earth spends part of their life cycle in the soil, and 1/4 of all organisms live in the soil permanently. To live permanently in the soil, the organisms need to get carbon from somewhere. And the only way carbon can enter the soil is through organic matter, which is mainly produced by plants. And we are going to discuss how carbon moves into the soil in much more detail in this presentation, as this is what really drives soil health. So all the organisms in the soil come together like a pyramid. The bacterial biomass at the bottom of the pyramid is about 1 to 2 tons per hectare. And the microbial biomass here can represent up to 1 to 4% of your soil carbon. The smaller the animals, the more abundant they are in the soil. And in reverse, the bigger the animals, the less there are in a handful of soil. And all these different organisms in the soil come together as part of the soil food web. So the typical diagrams usually show linear relationships, but it's not linear. There's organisms overlapping each other and finding very special niches. And this is really important to understand how these different animals or different organisms within the soil interconnect with each other. But before I go on to that, I'd just like to take a sidestep and look at some of the work that we've been doing, looking at how land uses can drive the different soil biology. And this is some work I was involved with the University of Queensland in the area where I live, up in North Queensland. And in this region, there are 4 main types of land uses. There are natural rainforests. Get my pointer. There are semi-natural areas, pastures. There's sugarcane and bananas. And the soil under agricultural land uses, the red and orange bars here, have about half the microbial biomass of the less intensive land uses, either the natural systems or the pastures, the green bars. And we know this is primarily due to the way the soils are managed. Because if we can compare wild bananas which grow in the rainforests, which are naturally there, and they have about the same amount of biomass as that of of the rainforest and pasture soils, twice as much biomass as compared to cultivated bananas. But as well as looking at the mass of organisms, we can look at who is there in the soil, either as an individual organism or as a community of organisms. It's a bit like looking at a population in a rural town. They can either exist as individuals or a community. In the charts, each dot of these dots represents a community of soil organisms from the different land uses. And we looked at 20 different communities from each land use. Those communities that come together from the same land use tend to cluster together compared to those from the different land uses. So we see all the red dots from bananas very close to one another. Sugarcane is separate, and then the natural systems. And they find that's the same for fungi as well as bacteria, that the communities in each of those land uses tends to be very similar to one another, but quite distinct from the different types of land uses. But as well as looking at the communities of organisms, we can look at individual organisms like Fusarium oxysporum group, which is a big problem for our banana growers, as this group contains the pathogen that causes Panama disease. So what we find in natural systems is that there's not a lot of Fusarium in those soils, but as we get into agricultural soils, it jumps up quite dramatically. And in our intensive banana soils, there's nearly an exponential leap in the amount of Fusarium that dominates those soils. So what I'm trying to say here is that how the land is managed, how we use it, really changes the organisms in the soil and really changes not only the individuals, but the whole community as they come together. Let's get back to our soil food web and how the different organisms interact with one another. And let's start off by looking at a citrus growing system. And I guess I'm using this kind of system as a worst case scenario from what we've learned within bananas. So I'm going to track how the carbon moves through the soil food web and impacts soil health. And if your farm looks something like this, I'm sorry, but it's not going to be a pretty story. So carbon needs to enter the soil food web. And again, this is work we've done on bananas, so I can only speculate how it may work with citrus, but I'm expecting it to be much the same. Plants get their carbon via photosynthesis from sunlight and carbon dioxide in the air. They fix that carbon and it can enter the plant and come into the roots. In this system that I showed previously where the soil is bare, the only carbon entering the soil is from the roots of those plants because there's nothing else there. So what we are doing is selecting organisms that are well suited to get their carbon from the roots of the plants. So for the microorganisms, we're selecting parasites and pathogens, so like Phytophthora, Fusariums, that can get their energy from the roots of the plants. Then as we move down the food web and get into bigger organisms, we're still selecting organisms that can get their energy and carbon from those roots, such as nematodes, plant parasitic nematodes that rob the plant from that carbon. And then we keep going down. We keep selecting all the organisms, the insects, and that can take carbon out of those plants. So we get a little bit of nutrients being recycled, but a lot of carbon dioxide produced. We can actually map how that moves through using nematodes. So if we have a carbon resource, what we find in this system is that most of the energy goes through the parasites. It's a little bit that goes through bacteria, it's a little bit for fungi, and there's a few predators, but not a lot. So most of the activity in the soil is supporting the activity of parasites. And from some of the work that we've done again with the University of Queensland, is that we find if we change the amount of biomass in the soil, that is the amount of living organisms, and we go backwards and we lose those organisms, we actually increase the probability that pathogens can colonize the soil. So if we get a 50% reduction in soil biomass, we get a corresponding 50% likelihood that a pathogen will colonize that soil. And these are the things that tend to destroy that bioprotection, where we have a monoculture with waterlogging, drought, heat, and salinity. We keep the soil bare. If it's tilled a lot, we have poor drainage and excess fertilizer and pesticide inputs, and if that soil remains acid. So that's the bad news. So let's go back to a better news story and have a look at another different type of citrus system, such as this one, which is breaks up that monoculture by having more plants in the system, like these vegetative ground covers. If we go back to our soil food web, it looks a lot different. We again have our plant that captures carbon through photosynthesis from sunlight and carbon dioxide, but instead of all the energy coming into the soil through the roots, we get this dead and decaying organic matter. And this means that to break that down and release the carbon, we have the microorganisms as decomposers of that organic matter. So the fungi and the bacteria are getting it from that decaying organic matter. And then as we go down our food web, instead of things still getting their roots, they're feeding on these other organisms. So the microfauna, like the nematodes and the protists, are all feeding on bacteria and fungi to get their carbon. And then the bigger organisms, the mesofauna, are again feeding on the nematodes, they're feeding on the other fungi. And so instead of having a straightforward, simple food web, we get this highly connected food web where all the different organisms rely on each other for their food. And it's not as simply just coming from the plants. And again, we can map this, and we have done this in bananas. And what we find in systems where we break those monocultures and have more plants is we get much less parasites, and the energy coming through the parasites is greatly reduced. And we get bacteria and fungi having a greater role in moving that carbon around the soil. And we also get a much bigger pool of predatory nematodes that feed and control and regulate the population of different organisms in the soil. So going back to what we found with our work and looking at the changes in biomass, is if we can double the biomass in our soil, we then halve the likelihood that a pathogen can colonize that soil. So this is the green area that we're moving into, where we change the biomass by doubling it, and we're halving the likelihood that that a pathogen like Fusarium can colonize that soil. So the things that really build this bioprotection, having those diverse organic matter inputs, having mixed cropping, having vegetation cover, minimal tillage and soil disturbance, good water drainage, balanced nitrogen inputs, and organic fertilizers source, again, which are balanced, and having a neutral soil pH. So I just wanted to reiterate some of this, if I can, is that then looking at what this looks like in pictures. So we started off with our monoculture with very little diversity. So the soil bioprotection is probably fairly limited. But if we maintain soil cover, we add mulches like these amendments, and we get that vegetation cover, then we start to really build these biodefenses. So I wanted to go on now of why things like ground cover are so important. So I was showing how we could map the different flow of organic matter, but we can also map the organisms in the soil. And in this case, we've got our pathogen, which is Fusarium, the orange dot in the center, and how it interacts with different organisms. So all the black dots represent a different organism in the soil. The blue bars are things that increase as our pathogen increase, and then their red lines are things that are helping to control our pathogen. And in this case, we've got about 10 different organisms helping to control our fusarium, which is about 60% of the organisms that interact with fusarium. But if we go to a bare soil, we find that we have our fusarium again in the soil, our pathogen. There's only one organism that's trying to control it. So we've lost all our regulation in that bare soil compared to what we have in a vegetated ground cover. So if we increase our organic matter, we increase the microbial diversity, we get less pathogens and less disease. And we've shown this again in surveys with banana growers, where we're really looking at trying to get a big picture of soil health. And we're looking at the relative abundance of fusarium in different soils, different systems. Where we have low ground cover, like in the system at the top, we find we have a much higher abundance of pathogens of like fusarium. Where we have a high ground cover, greater than 66% of vegetation cover, we actually have a much lower relative abundance of fusarium. But it's not just the pathogens. We get a whole lot of changes in chemical and biological properties. And they're currently developing a kit that allows us to test these in any agricultural system and to pull these different measurements together. Moving on, just to reiterate about some of the organic matter, how important it is. Organic matter is not the same thing. So while I've said it's about 5%, about 70% of the organic matter is humus. There's a bit that's 15%, which is like charcoal, and there's about 15%, which is a labile fraction, either fresh organic matter or living organisms. And it doesn't stay the same. It undergoes transformations. Each time there's a transformation, there's a little bit of carbon dioxide that's lost. But what we find in the soil, that it's largely protected, that it's in different areas. It's not just a single layer. It's protected in the soils. And the more we disturb those soils, we expose that from its protection and allow it to be degraded and more carbon dioxide being lost. Similarly, if we flood this system with nitrogen, then it just allows the bacteria to help degrade that carbon and releases the carbon dioxide. So what happens over time when we add organic matter? It starts off with a lot of nitrogen compared to carbon, so a low carbon nitrogen ratio. Those easily broken down nutrients are released and lost, and so then we end up with more carbon and less nutrients. So we go from labile products to recalcitrant protected carbon. Now organisms go from what is termed copiotrophic organisms, fast growing, fast multiplying, that like the food, fast living organisms, to oligotrophic organisms that are slow growing, and they need to use tricks to be able to survive. So they use all these different mechanisms like producing antifungal metabolites and help control the different organisms. So we move from predators, and then we also get hyperparasitism. Nitrogen is an interesting component of soils. We need nitrogen to grow plants and grow crops, but the more we put on, the more we select some of these pathogens in the roots. Like this is what we found with bananas. We're getting more pathogens in the roots. pH has another really dramatic impact on the diversity of organisms. We look at the bacterial diversity around pH 7. If we change that and decrease the diversity, then we really drop off. If we change the pH, we really decrease the diversity of soil organisms. Similarly, with fungi, we have a maximum diversity around pH 6. If we go either side, if we become more acid in the soil to pH 4, we decrease that diversity. And similarly, if we go higher and go towards a pH 8, we decrease that diversity. So what I'm trying to do with all this is trying to pull how we can pull all this together. So on the one hand, we have our management practices, how we fertilize, tillage, we use vegetations, amendments, pesticides, and water. And then we see what services these different management practices may have. And we usually try and just see a direct link, but it's not a direct link. There's quite a convoluted pathway that these practices get into what we see as soil services. So at the bottom of it is the physical properties, the chemical properties, and at the top, it's predators that regulate the soil services. And I think Emily has another poll that she's going to launch now. So the question is, which of the important soil services, these purple ones on the right, do you think is most important for your farm? Is it organic matter degradation, nutrient recycling, soil structural improvement, disease suppression, degrading pollutants like pesticides in the soil, or greenhouse gas regulation? If you're able to answer the polls, a few answers coming in. Yeah, so for most people, it seems to be nutrient recycling is really important, followed by the organic matter degradation and soil structural improvement with disease suppression down the bottom, and water quality, which is one I forgot to mention, also down there. So that's really important, is people really need to want soil health to provide them with more nutrients. And so what I said right at the beginning, that soil health is intergenerational. What you're trying to do is hand on the soil in a better condition. So there's some figures that say that the way that we are managing the soils is that if we use conventional management, around 16% of soils only have a lifespan of around less than 100 years. So some of those soils are going to be lost, unsuitable for agricultural production in less than 100 years. But if we change our practices and undertake some of these more conservational type practices, like adding organic matter, getting plant diversity, managing the pH, we're likely to drop this, that only 7% of soils will have a lifespan of less than 100 years. And we greatly double how long those soils can then be productive agricultural soils. So if you want to hand your soil on to the next generation, into your grandchildren, I suggest that we really need to look at how we manage those soils. So, and we can do this by understanding what soil type we have. We look at the constraints, and then we apply the management that's appropriate to overcome those constraints. Look at our soil services that you've already said that you're really interested in, like nutrient recycling. We analyze to see whether that's working, and then keep improving. So as a conclusion and to wrap up, I hope that from this presentation you realise that soils contain millions of organisms that rely on plants for their carbon, either from decaying organic matter in the best case scenario, in the worst case scenario, they're going to rob it from the roots. Farm practices have reduced soil microbial biomass, and we showed that in our banana system where it's half of what we find in natural systems. So this then makes it easy for plant pathogens to dominate. And building soil microbial communities through farm management builds bioprotection against soil-borne diseases and restores a lot of those really important ecosystem services. So just acknowledge everyone that has contributed to this, my colleagues with Department of Primary Industries now, need to update, and also from the University of Queensland. And thank Andrew and Emily for inviting me to come along today.
Thanks so much, Tony. We just had one question from the chat, and that's from Jude, who's asking, so what are parasites in the soil system?
So some of the parasites for citrus system would be nematodes, and citrus nematode can be a big problem in many places. Also, we have root knot nematodes and different. So there can be different parasites within citrus systems that I'm aware of. The pathogens, I think that's something that Nerida is going to talk more about. So I'll leave Nerida to talk to that.
Jude also just had a comment that there was still some interrow grassing in that photo you had of the bare paddock. So would you consider, I mean, I realize that it's probably not the most extreme example of a bare citrus paddock, but would you still consider that to be enough to improve the variability of a citrus paddock?
Looking at that photo, and I think that's an excellent question, that's probably under that 35%. So what we're really looking for is how do we get those the ground covers, the vegetation interacting with the roots of our crops, and that's what we're trying to do within the bananas. So trying to push that vegetation cover as close to the roots so that we can get those interacting the differences between the root systems, the organisms that live on our ground covers that are typically beneficial ones, interacting with the roots of our crop so that we can get those benefits and controls. So I don't think in that strip it's quite narrow in the middle. It might be enough to slow some of the erosion down, but I don't think it's enough to really drive that soil biological improvements. And I guess the way to really understand it is to measure it and see what's going on in different amounts of systems.
Nerida is our citrus pathologist at New South Wales Department of Primary Industries and Regional Development. So yeah, Nerida handles a lot of our plant health diagnostic service inquiries. If I'm asked to go to a farm and I know the problem, I'll send them off to Nerida's lab at EAMI. So Nerida, you're welcome to share that screen.
So that was a great presentation from Tony about soil health and what you need to think about. So I'm just going to take you into some more citrus specific areas relating to soil health, but you need to keep remembering what Tony has said about those basic principles. Well, it's not really basic. The soil's very complex. So just a reminder, I'm Nerida Donovan. I'm a citrus pathologist with the New South Wales Department of Primary Industries and Regional Development. I run the citrus pathology program. So we work on prevention, detection, eradication, and management of all diseases of citrus, exotic and endemic. Not so much work in soil health, but I have worked in soil health in the past with Tony in vegetables and back in my past life in grain crops. So a healthy soil, to remind us of Tony's talk, that implies that we've got some ecosystem stability, diversity, maintenance of soil processes or soil services, as they're called by Tony, and resilience in response to disturbance or stress. So what we've done is we've taken this natural environment that had been developing over how many years, thousands of years, millions, and we've changed it. We've created a monoculture. We may be doing some cover crops, but it's still not the same diversity that they had before. But eventually the system will get into an equilibrium, and that's what we want to work towards. So ecosystem stability, and we want to have that functioning to be doing everything that it needs to be doing. So if I may, I'll spend 2 or 3 minutes just a little bit of a history lesson as to why we're actually doing what we're doing at the moment. So if take you back to 1861, and there were citrus orchards around Sydney, of all places, and they had widespread tree decline. They didn't know what Phytophthora root rot was, of course, but looking back at the records, they've got great descriptions of the symptoms, and we now know it was Phytophthora root rot. So what was the response for industry? They introduced sour orange rootstock. They started using sour orange more often. But then because overseas where they'd had similar symptoms, sour orange was quite a successful rootstock. Okay, so that's an important point. The problem was, at some stage, we've worked out before 1874, citrus tristasia virus was introduced to Australia. So the late 1860s, the 1870s, industry said quite clearly, no, sour orange rootstock is starting to decline. We've got this quick decline. They didn't know about viruses back then, but we now know it was Citrus tristase, a virus, quick decline with oranges and mandarins on sour orange rootstock. So discontinued use of sour orange, and we haven't really used it since, even though it was used and is still used because of Phytophthora issues overseas. There was, any chance I get, I'll talk to you about, I'll go rogue and increase the message. So budwood was sourced from thrifty trees, and there was issues with trueness to type with the post-war boom for budwood. And so the industry response was to establish the Cooperative Bud Selection Society. So buds were only selected for propagation from trees that were healthy. And they had to be healthy for 3 seasons and inspected by the Department of Agriculture. So the industry started the society, but it was with a government grant and there was government support. But again, after a few wet seasons, Phytophthora reared its ugly head. There was root rot and collar rot. That was labelled Phytophthora in 1941 as the cause of the tree loss on sweet orange and rough lemon rootstocks. So the industry response was to increase the use of trifoliate rootstocks and hybrids. And then Trifoliata Improvement Committee was established. The problem was there were increasing reports from industry that trifoliata wasn't all created equal. So there was variability in what the trees on trifoliata looked like. That was discovered that in 1947, they discovered that, that Exocortis, they worked out it was graft transmissible. And so some of the trees had Exocortis, but some of the trees didn't. So they didn't know what viroids were back then, but they were describing the symptoms and they worked out what was going on. So the industry response was to develop the parent tree registration scheme. So I just wanted to go through that to show you why we're doing what we're doing. We're using trifoliata rootstocks and their hybrids because of Phytophthora, but we recommend using budwood from health tested trees so that they don't have the viroid, because trifoliata might be resistant to Phytophthora, but it's very susceptible to Exocortis viroid. So it shows when you're making your rootstock choices, you're not just matching it to the soil, you're also thinking about these pathogen issues. So Phytophthora, the soil inhabiting organisms, they're called fungi, but they're not actually fungi, they're oomycetes. But in the general vernacular, people lump them with fungi. They attack under favourable wet conditions, root, stem and fruit decay, but we'll focus on the root and the base of the trunk decay today. And the spores can be splashed onto the trunks, leaves or the fruit. And ants and snails can also move the spores. So the first sign is the gumming. You'll see the gumming around the base of the trunk or maybe under tree guards. The affected bark is wet and soft and discoloured. And with time, the bark can dry and crack. So with mature trees, normally they would recover and it would heal over, it would harden. But it can, particularly with younger trees, with smaller trunks, you can get them ring barking and killing the tree. Okay, root rot, you get the decay of the feeder roots because not as many nutrients and water is going up to the canopy. You get canopy thinning, twig dieback, the new foliage might be weaker. If the roots die faster, then they can be replaced. If the soil stays wet, then the lesions can form on those more structural roots, the permanent roots as well. But as you can see, it looks like the dying tree, the defoliating tree with both. And I'll just raise that there is a fact sheet produced by this IPDM project on the New South Wales DPIRD website, Phytophthora in citrus, and it has this disease cycle. It's simplified. There's a bit more to it, but this is the key message that you need to know. So management of Phytophthora root and collar rots. Yes, we've talked about resistant rootstocks, and you can see the different tolerance in that graph there. So it's from most tolerant with citrus trifoliata, and it's an old graph. I couldn't figure out how to change it from P to C. And all the way up to the least tolerant, which is sweet orange. Now, we did have a survey that was sent out to everyone about chemical management. But with your survey, if we go into the results, it said, it's a bit hard to see, there is application of phosphorus acid at least once a year. Some people do it more regularly, but when you drill down, why do you apply it? A lot of people were applying it because it's part of the orchard management program. So I'll go on to this in a minute. Other ways to manage is ensure the bud union is a minimum of 20 centimetres above soil level. You buy healthy nursery trees, ensure good drainage and irrigation management, healthy soil microbial biomass, manage the ants and snails so they're not moving the spores around. The placement of the sprinkler. So you can see in that photo, there's a sprinkler right against the trunk, and that constant wetting on the trunk has caused that tissue to soften and become more vulnerable to attack from organisms. Monitor under tree guards, just make sure there aren't things coming up, and then we'll get back to the chemicals. But I wanted to just mention, so there's resistance in the trifoliata, which means there's Phytophthora there, but it's not causing the rotting of the roots or the trunk. There's citrange where the hybrids where you've got tolerance. So there might be some rotting of the roots, but they're replacing it faster. So there's less of an impact on the tree. With chemicals, phosphonates are quite a complex mode of action. They don't really know how they work. They're the preferred chemicals for Phytophthora management because there is that complex mode of action. So there's always thought to be a low risk of resistance developing. But there's reduced efficacy has been reported in citrus, particularly in the last 5 or more years. Further research is needed in this space. Like, they need to do more work. A lot of the most recent paper out of South Africa in last December was talking about, it did a nice review on this. So it's just cautioning about careful resistance management. Yes, it may be something you've always done. You may see an impact on tree health, but with anything, it's got a cost. So you need to carefully review it and not do it just because you've always done it. And you've got to take care with resistance management. But there's also a situation where if you haven't got ideal management conditions or best practice management, you can overcome the tolerance or overcome the resistance in the rootstock. Say a case of superinfection, so the conditions really favour the pathogen, you can start to have more of an impact, particularly with tolerance. There'll be more rotting, and then the roots won't be replaced as fast as they're dying, and so there'll be more of an impact above the ground. Okay, misidentification can happen with anything, and there's so many things that can look like a dying tree. Okay, so say there's some photos there of termite damage, citrus tristeza virus, and that's quick decline. Exocortis viroid can actually cause that peeling that can look a bit like the dried and peeled and cracking Phytophthora damage. And there's gumming. Different viruses like cirrhosis virus can cause gumming on the trunk of the tree that can look similar to Phytophthora, but you look at where it's located, and you just got to delve a bit deeper. So you don't see a dying tree and think Phytophthora, particularly if you've got good conditions, good irrigation management, and you're on trifoliata or citrange. Okay. So just quickly on a couple of other things, there's soil salinity. Salt sensitive, citrus is a salt sensitive crop. You obviously get direct absorption of the salt through overhead irrigation, but also through the roots during normal water uptake. Accumulation of toxic levels of salts in the leaves can lead to leaf burn and defoliation, so the leaves fall off, and long-term yield decline. Rootstocks can be either excluders or accumulators. They have different levels of tolerance to salt. So that's why you'd also look, if you've got quite salty irrigation water or a salty environment, you'd have to use certain rootstocks that were more tolerant, like Rangpa lime or Cleopatra mandarin. It's about getting that balancing act of what your issues are on your farm with your rootstock choice. Changing from overhead to drip can reduce the impact of saline irrigation water, but if you in a low rainfall environment, the salts can still build up on the edge of that wetting zone. So you may need to use the overheads to leach the soil and push the salts down. Reduced effectiveness of Phytophthora resistant rootstocks in saline soils. So you may get Phytophthora issues even on, say, citrange or something, if you're in a saline soil. Compaction and soil aeration caused by heavy equipment, the traffic, over irrigation, even the weight of the tree is heavy. Soil structure can be destroyed, reducing that pore size. So Tony spoke to you about the balance with the water and the air and the soil. So anything that compacts the soil, it means the tree has to use more energy to grow. The roots have to use more energy to grow through the soil. And the root growth is particularly reduced when the airfield porosity is less than 15%. So you've got the air issue, but you've also got the issue of the energy required. And there's increased susceptibility to Phytophthora and other fungi. So if you've got a heavily compacted soil, you might have citrange, but the Phytophthora might still get in there, still win. Okay, nematodes. I felt cautious about talking about nematodes in front of Tony. He's nematode man. So they can cause slow decline, reduce tree vigor, yellow foliage, sparse small fruit size. Again, trifoliata, the citrange, Swingle citramello, they've got resistance to nematodes like they do to Phytophthora. You could use biofumigant cover crops. So Tony's encouraging you. It's great to use more cover crops, but why not kill two birds with one stone? If you've got a nematode problem, you may as well plant those sorts of cover crops. But again, there's that issue. Soil salinity can increase the population density of nematodes by increasing egg production. Salinity in this study I found had no major impact on nematode resistance of rootstocks. But nematode damage can increase the adverse effects of salinity on citrus trees. So that it's so complex. There's not independent issues. They are impacting each other. Sudden death is where apparently healthy trees, often with a good crop of fruit on them, suddenly wilt and die. You get black rotting structural roots at depth, dry brown discoloration that extends into and across the butt of the tree. You can see that with the cross section of the trunk. And some of you may recognise Lou Revelant, who's crouched down under the tree in the photo. The Griffith people might. So you could also get ink-cap fungus, Coprinus species at the base of affected trees, mainly in autumn after humid weather. And there is controversy over the role of Fusarium solani. So overseas, some people say that sudden death is linked with a certain Fusarium species. But in Australian studies, they didn't find one main cause of sudden death. It was more a case of it was a physiological issue, and whichever organism happened to be dominating the microbial population got in there and took advantage of the situation as a secondary invader. So intermittent periods of waterlogging and poor aeration led to a weakening of the root system, invasion by those microorganisms, gradual death of the root system, and then tree collapse. So you normally see sudden death. It affects trees of all scion varieties on tri, chorizo, and Troia citrange rootstocks. Can occur at all ages, but mainly 7 to 15 years, and all soil types, predominantly heavy soils. So maybe that's been skewed with the rootstock types, because if you're on a heavier soil, you're more likely to be on tri or citrange rootstocks. So that might be skewing the data there with what we're seeing sudden death on. Okay, you can reduce those losses by careful site selection, surface and shallow subsurface drainage, irrigation monitoring. So just the usual things that you do, even application, and mounds. I forgot to mention mounds with Phytophthora as well. So with anything, you can improve drainage, you can improve the depth of the good soil by mounding. Replant disorder occurs in many tree crops. Citrus trees may grow poorly when replanted on soil that's been under citrus cultivation previously. Now, we've tried to look at this, but they've tried to look at this all over the world, and it happens in apples, it happens in other tree crops too. There's lots of hypotheses, but it does seem to be site specific. And so when I did this study at DET, and it was about reduced diversity, there seemed to be bacteria dominating the fungi in that soil microbial system. So it is quite site specific. So we do recommend a fallow period or some cover crops before you replant the citrus to try to avoid replant disorder. I think we've talked a lot about organic matter. Organic amendments can be applied to your soil. If you haven't got that full cover crop while you're trying to grow one, you can add organic amendments to get improved tree health and pathogen suppression. But again, you're not going down the street and buying a bottle of Coca-Cola where you know what you're going to get. There can be so many differences in the quality of amendments, and there are different site factors that come into play. But for Phytophthora, there's many studies, and they've known this for many years, this reduced issues with Phytophthora in tree crops. I think the original work was avocado, with using organic matter. Okay, and it can have an impact on nematodes as well. Cover cropping, I think we've covered this with Tony talking about how try to make sure the cover crop and the citrus roots do connect to get that benefit. It's quite common to get bare earth or just a small strip of cover crops in citrus, so you may not be getting that benefit. And some of the drawbacks that people mention are risk of frost damage and harbouring insect vectors and pathogens. That's not so much of an issue for citrus though. If we got something that had a vector that also was on weeds that spread disease between the citrus and the weeds, that would be more of an issue, but we don't have that issue in citrus. So if you see that written, it's not so much of an issue with citrus. There are microbial biocontrol products that you can use. Again, they can be very different, but there's been some success in citrus against Phytophthora with Bacillus and Trichoderma. And beneficial soil organisms. So citrus are highly mycorrhizal. That's one type of beneficial organism. The hyphae explore the soil and they return nutrients back to the root. They can also be an improvement in water uptake and photosynthesis, better growth, and improve soil structure, because actual hyphae out there help hold the soil structure together, provided it's not compacted. It's nice to hold these nice loosely formed PEDs together with hyphae. It's a nice balance.
I was going to say, we've just got a couple of questions in the chat from Noel Ainsworth, which I think Tony and Nerida might both be able to comment on. With citrus being such a surface feeder and voracious feeder, are you better off keeping the area to the drip line covered in mulch rather than letting grasses and other vegetation encroach there?
Yeah, I'll tackle that. But start off, and I think this comes to what Nerida said earlier. It really depends on your environment. If water is limiting, the most limiting things, then you don't want things that are sucking up your precious water. In our environment, water isn't a constraint. It's too much water. How do we get rid of it? But I think there's also, so in an environment where water is limiting, then maybe you're better off with a mulch, an organic mulch rather than a living mulch. But having also said that, there may, when you've got a living mulch vegetated ground cover, they may take up some nutrients, but those nutrients are then going to be tied up within those plants. So as they die down and decompose, then they're recycled. and also having bare soil exposes the soil surface to the sunlight, and that's more drying of the soil. You'll lose more evaporation through that than you will through having a ground cover over the soil and shading it, which is the experience that I've had with comparing ground covers and bare soils, is that it tends to dry out a lot more in the bare soil than it does one with a living ground cover on it. But yeah, it's to get a ground cover established requires water, and if water's limiting, then an organic mulch, organic amendment type mulch, better option, I think.
There's just a couple of more questions. Any tips on basic soil health improvements for conventional growers on drip irrigation? You might not have so much experience with drip irrigation, Tony. That might be Nerida's area. No rain makes it hard to maintain mid-row crops.
I know at Dayton they use drought-tolerant rye. They have in the past, but it's only in a strip down the middle.
With the ground cover one, I think we'll find in there's a lot of work in vineyard systems where they grow different ground covers in the interrow at certain times of the year, allow it to mulch down. So when it is wet, they can get those crops in and then mow it. We're using a side throw slasher or something so that mulch is around the base of the plants to protect it later.
Thank you, Tony, for answering that. Look, I think we'll wind it up there for the day. Look, I thank you all for participating in this webinar of the IPDM Extension Program. Special thanks to Dr. Tony Pattison and Dr. Nerida Donovan for sharing their knowledge with us and taking questions from us. Their emails are up there. I'm sure they'll answer any questions from your participants if you wanted to contact them. I thank Emily Pattison for co-hosting today's webinar. I just want to remind everybody that this was an outcome of the IPDM Extension Program that was funded by Hort Innovation, your citrus levies, and also the collaborating state governments. And Biological Services and Riverina IPM were also a big part of that extension program.